Um, that's a good question. I don't think they should be in quotation marks. Yes, yeah. They're real. They're very, very real. But yeah, so all this chapter is fatty acid catabolism. How fats are digested, mobilized, transported. We're going to talk about their oxidation, which we'll see feeding in. Again, we're going to talk about acetyl CoA, and then we'll talk about the ketone bodies. So if we have no glucose, we have no carbohydrates. We're going to see that when we burn fats, we're going to end up producing three specific ketones that will be used for fuel in the brain. And let's see here. There we go. Okay. So, we're very human-centric here. So roughly a third of our energy needs come from triacylglycerols. If we look at the heart and the liver, we really burn a lot of fat in the heart and the liver. So, that's like 80% of our energy needs will come from that. If we think of all the little animals that are waking up, such as the grizzly bears living out. Okay, no grizzly bears here. But during hibernation, animals will rely almost exclusively on fats for their source of energy. So metabolism slows down, but I think there are actually some cams that are centered in Alaska, if you're ever interested, and they have like the fat bear contest. So they have these cameras in different territories of various grizzlies, and they kind of you take a bet. Now, I don't know if people actually bet on them or not, but you vote for who you think is going to be the fattest bear by the end of the summer, and when they go into hibernation. And since they're not eating anything else, they just burn the fat for fuel during hibernation. So there we go. And there we have not a grizzly bear, but a black bear, which I'm going to break with tradition here and tell a story right up front. So I grew up in rural West Virginia, right? We have bears around. Never saw a bear. The closest I came to seeing a bear, I think I was in graduate school and I was driving through the interstate through rural Maryland at this point, and there was like a little blob, a half a mile away off the interstate. I'm like, oh look, I think that's a bear. So a couple of years ago, we had shrimp for supper, and it was garbage night. So I put the shrimp shells in the garbage outside my garage here in suburban North Carolina, and late that night, I hear something kind of bumping up against the trash can. I thought it was like a raccoon or something trying to get in there. And I look out, and and then like it's knocked over, and I'm like, it wasn't the wind. And it's like maybe it's a raccoon, or maybe some kids were being all crazy, something like that. So I don't think much of it, and I start to go back to bed, and I hear thump, 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 and I was like, oh, I'm gonna go scare that raccoon off. I look out the window, and that raccoon is probably about 400 pounds. It's one of the largest black bears I had ever seen. It was huge and very healthy looking. And so then, of course, I go to the garage. So I'm about like 8 feet away from it, looking through a glass window at it. And I'm like, interesting. It's like, well, what do you do about a bear in your trash can in suburban North Carolina? Yeah. So, I was like, well, I should maybe call someone, and actually the sheriff sent like a patrol car out like a long time later. I was like, oh. So I had to go outside and say, I think he went around the house. But turns out this bear had actually been kind of going through the neighborhoods, and there were other people who captured him on like a ring cam or something like that. So it was an easy source of food. It's not like they need them. Yeah, and it was just, I found it very strange that as much as I grew up in bear country, okay, not really bear country, but relatives actually go hunt bears not too far from where I grew up. But my closest encounter with a bear is in the suburbs. So, but yeah, this bear had clearly learned where the good stuff was, and I was like, hey, boy, he loved those shrimp shells. I mean, they were wrapped up in foil, and he managed to open up the foil. I think actually it was a she. She managed to open up the foil and get them all out. So yeah. Anyway. And of course, the reason for that is, shrimp is very high in cholesterol, nice little lipid in there, lots of good fats in there as well. And fats provide very efficient fuel storage. If you go back to basic chemistry, you probably learn proteins and carbohydrates give you about 4 kilocalories per mole of energy, and fat gives you 9. So it's more than double the amount that's in there. The other thing is, the way we store fats, we can eliminate water, so we have a dehydration reaction, so we don't have water weight. So worry about for our storage, or the size of the wet molecules, or that's typically not too big. I always think about this as another thing from growing up in the rural areas. Every town that had at least a few thousand people had its own bottling plant, whether it was Coca-Cola or Pepsi or RC back in those days. They would just use local water, and then like the big centralized area would send out the formula to mix with local water to make that, because it was a lot cheaper to ship a truck full of the flavor compounds, what do we have in here, sugar, caramel color, phosphoric acid, caffeine, whatnot, and use local water, because the water weight was the heaviest component in there. So much like that economic thing. Boy, ran out of words real fast today. But storing fats as energy is very efficient way to have a lot of energy in there. So I mentioned before, we use glucose and glycogen for short to medium term energy. Our fats are long term energy, right? So it can take months to use it all up. Very good storage, very slow delivery in there. Okay. If we take a look at everybody's favorite picture, white adipose tissue, the white parts in here are fat droplets. This is all triacylglycerol, and then like the other little things that are stained in here are other parts of the cell. So I think you can see some, is that cytoskeleton maybe in there or something, and we have some muscle fibers, I believe, over here. I forget exactly what else is around here, but we got these tiny little nuclei. But our adipocytes are just these massive fat droplets with the rest of the stuff kind of crammed into the side around it in there. Okay. All right. When we eat fatty acids, so I was gonna say, I guess there's a little bit of fat in my beef jerky, but it's pretty lean meat, and then I had a roll, and so I'm just having nothing but sugar. I'm having a low fat breakfast, I guess. But if I'd fried up some bacon and gobbled that down, our fats would be absorbed in the small intestine. Basically, because fats are hydrophobic, they don't well. So bile salts from the pancreas and gallbladder will emulsify them and allow them to form micelles. So we essentially get a coating that is hydrophilic out there. We'll see some various lipases. So again, lipids, lipases are going to be things that degrade triacylglycerols and cleave them into free fatty acids. They will get broken down and taken up by the intestinal mucosa and then converted back into triacylglycerols. And these can be incorporated into various small molecules. We typically start with chylomicrons. They are the largest of these small molecules that contain a lot of fat, a lot of triacylglycerols in there, and then what we call apolipoproteins. If this were another level up of biochem, you'd have to know all the apolipoproteins, but we're just going to focus on a couple in there. But the apolipoproteins are kind of also on the outside, so they interact with various receptors to promote the reactions that we want to see with these chylomicrons. So chylomicrons, you can see we kind of draw on our apolipoproteins on the outside here, but the chylomicrons will move through the lymphatic system in the bloodstream to the various tissues, and then we'll have lipoprotein lipase, which gets activated by this ApoC2. So apolipoprotein C2 is important that way. We'll convert the triacylglycerols to fatty acids and glycerol, which can then be uptaken into the adipocytes or other organs to be used as fuel. Adipocytes for storage and other organs for fuel. And we can generate ATP and CO2. All righty. So myocytes are adipocytes. So muscle cells, primarily think heart muscle, because we get a lot of our energy in the heart from burning fat. Okay. So let's start off just kind of looking at this in a little more detail. I always joke that one of my end of first year grad school biochem exams was basically a rat eats a piece of meat. Describe. That's a little bit of hyperbole. It was really a rat eats a piece of meat once it hits the portal vein. Describe. So once we get into the liver, we're not going to be too concerned about uptake in the gut and all that. Just know that we do have lipases that will cleave these things into manageable molecules that can be taken up in the stomach, but they eventually all go through the portal vein in the liver. Okay, so chylomicrons are our biggest and fluffiest of the lipid transport molecules in there. They will have a lot of triglycerols and cholesterol esters in the center, with some cholesterol on the outside and some phospholipids, and some of these key apolipoproteins. We'll see B48, C2, and C3. Now, the naming of the apolipoproteins, it's B48, it's named 48 because of its molecular weight, C2 and C3 because there are variations on that. So it kind of depends on, the naming is not consistent across the apolipoprotein families because of different labs and investigators who discovered what they did. All right. But chylomicrons are our first and biggest fluffy molecules. They're the least dense. They're also physically the largest. We will also talk about low density lipoprotein, very low density lipoprotein, high density lipoprotein. There's an intermediate density lipoprotein as well too. But as we go from lower density to higher density, we decrease the quantity of lipid and increase the quantity of protein, which is what affects their density. Many hormones will trigger the mobilization of stored triacylglycerol. We can activate these various lipases by glucagon and epinephrine. So epinephrine, you might think, well, if you need energy really fast, right? Well, it's also going to mobilize some of those fat cells to send triglycerides to the heart to use for energy. Although we're going to get the bulk of our immediate energy from sugar release in here. Glucagon is similar to epinephrine in its actions, but it's also a response to hunger. So hunger arises from a low energy state. So glucagon can bind to its receptor. Again, we see a G protein coupled receptor, right? GPCR, which will activate adenylyl cyclase. We see our second messenger, cyclic AMP. It's like a review of chapter 11, right? Okay, we activate protein kinase A. But then within our adipocyte, we're going to see these hormone-sensitive lipases. Okay, so hormone-sensitive glucagon is a hormone. We go through a signal cascade. We activate hormone-sensitive lipase. It becomes phosphorylated. It will move to the fat droplet in there, and then in those regards, it will start cleaving our triacylglycerols, or diacylglycerol, into a monoacylglycerol and a free fatty acid. We can also see protein kinase A activates paralipin in the membrane surrounding the fat droplet as well too, which here we have our triacylglycerol lipase that will cleave a triacylglycerol into a diacylglycerol and one free fatty acid. So we basically have these, three fatty acids that are connected to glycerol, if you recall from way back in chapter 10, I believe. Yeah. So we're going to cleave one off with one lipase and then cleave the other two in half with another lipase. And eventually what we end up doing is we end up with three free fatty acids as well as a glycerol molecule, which can be used in a variety of ways. And then these free fatty acids can be released into the bloodstream, where they can go to where they're needed, most likely the heart cell and other myocytes, sometimes the liver. There are various fatty acid transporters. This is definitely an oversimplification because we don't just release free fatty acids into the bloodstream. They have to be carried by various carrier molecules, like our VLDL, LDL, HDL, all that good stuff in there. But once they get to their target cell and bind a receptor, they get transported inside the cell, and then they can get fed into the citric acid cycle. So they have to also get into the mitochondria as well, right? So we're not showing that on here. And once we're in the citric acid cycle, we start breaking down our fatty acids in 2 carbon units. So we have 2 carbon units. You might think acyl-CoA, acetyl-CoA. Are these basically acetyl groups that we're going to be making from that to make acetyl-CoA that can feed into the Krebs cycle and help generate energy. And we're going to look at palmitate as a model example of this as well too, our 16:0 simple lipid. Okay. All right. So how does this all work? All right. So just as kind of a refresher, this is our basic structure of a triacylglycerol. We have our glycerol molecule here. It's a little 3-carbon unit with 3 hydroxyl groups. We can esterify fatty acids on there. These are condensation reactions. So we're eliminating water to make these ester groups in here. And in this case, this is a mixed one because we put a bunch of different stuff on there. But typically we'll see 1 unsaturated fatty acid and maybe 2 saturated fatty acids in there. Okay. And unsaturated just means that we have all the possible hydrogens available in there. Unsaturated means we have double bonds in there. And recall our double bonds are almost always cis. Trans fats, not natural in organisms. We can make them. They also increase the shelf life of butter, but it's been probably about 15, 20 years since pretty much every restaurant stopped using trans fatty acids because they increase the risk of heart disease and plaque buildup. All right, so first we'll talk about the glycerol. A glycerol from the fats can actually enter glycolysis. Okay, so glycerol kinase will activate glycerol. It kind of is our investment phase. We're going to spin an ATP to phosphorylate it. So we end up making this dihydroxyacetone phosphate, DHAP, sounds familiar. So we can convert that into glyceraldehyde 3-phosphate. Our GAP3 phosphate there then is one of the intermediates, and that's right before you get to the payoff phase. All right. So basically, the glycerol kinase activating this will recover enough ATP. Basically, you spend 1 ATP, and you're going to get, I think, 2 ATP out of this as you go through the second half of the phase in there. And basically, this means that we can have a very small amount of anaerobic catabolism of fats. So we don't need oxygen for this to occur because we can actually take the glycerol and shunt it into the glycolytic pathway. All right, so next couple slides are just this in a little bit more detail. We start with our glycerol molecule. It's interesting. I don't like drawing it this way, but this is the correct way to draw it. I prefer that kind of 3-dimensional structure here, but now because we're in a pathway, we're going to change the way we draw it. We are going to spend ATP and phosphorylate it. So we phosphorylate carbon 3 and make our glycerol 3-phosphate. We're going to rearrange this. Okay, so here you can see we're generating an NADH. So already we're getting some effective energy back by making our reducing agent. Dihydroxyacetone phosphate. Okay, and then we're going to rearrange that. This may look familiar, triose phosphate isomerase from the glycolytic pathway, and we've now made GA3P, okay, or GAP, or however you want to call it, which is one of the intermediates in the glycolytic pathway. Okay. So we have kind of skipped the first couple of steps from glucose, and we've just taken the 3-carbon sugar and converted it into a metabolite of the glycolytic pathway. Okay. So if we're going to transport or attach phospholipids, we're going to need some conversion to fatty acyl CoAs. So our coenzyme A, which we talked about in chapter 16, we're going to talk about it in like the next 3 or 4 chapters in here. It plays another role. We are going to make this coenzyme A and have it interact with our fatty acid. So we've talked about acetyl-CoA, which is just a 2-carbon unit, but we could take, a 16-carbon palmitoyl and have, palmitoyl-CoA. So we basically just have this some type of fatty acyl-CoA, where our sulfur is going to be attached to the carbonyl carbon. Okay. All right. And this is a 2-step process because the first thing we need to do is, phosphorylate the fatty acid, and then that will make this a good leaving group so that our CoA can attack the do nucleophilic attack on the carbon in there. Let's say carbanion, but carbocation. All right, and then we'll release that phosphate group there as well. Okay, this is a very favorable reaction. So we'll see some nice release of energy there. Once we get the fatty acid to its target cell, so it's like, oh, my bacon has made it to my heart. Now what? We got to get it into the mitochondria because that's where the citric acid cycle is. So the fats are degraded into fatty acids and glycerol in the cytoplasm, but the fatty acids, the beta oxidation occurs in the mitochondria. And this separation helps separate fatty acid oxidation from fatty acid synthesis. All right. So we actually bring it into the mitochondria to get the energy out. When later on, when we look at fatty acid synthesis, we're going to see that takes place in the cytosol. So again, we have separated these to avoid a futile cycle. Okay. All right. Interestingly, there are multiple ways to get fatty acids into the mitochondria, and they depend on the size. So small fatty acids, will freely diffuse across the mitochondrial membrane. If you drink milk, you have a variety of fats in milk. Some of them will freely diffuse across because they're small. I think was it capric acid and lactic acid are small chain fats. Larger fatty acids, though, are too big to diffuse all the way across, and they need a transporter, and this is the acylcarnitine transporter. Like, wait a minute, what? So yes, we're going to acylate our carnitine and regenerate it to help get it across there. Okay. So our acylcarnitine transporter kind of works like this. Here we have our fatty acyl CoA, all right, and we have carnitine, and they're going to interact, okay, to give us back our coenzyme A and our acylcarnitine. Right. We then have this carnitine acyl transferase 1, which gets us across the outer mitochondrial membrane. Right. Then we have a transporter in the inner mitochondrial membrane that will bring that into the matrix. And this one is an antiporter where carnitine from the inside flows back out as the acylcarnitine flows in. All right. Once in the matrix, we have this carnitine acyl transferase 2, which is going to remove the carnitine from our fatty acid, and we regenerate our fatty acyl CoA and generate free carnitine, and that carnitine gets transported out as the fatty acyl carnitine comes in. So carnitine is acting like the cab. That was my mnemonic. I thought CA carnitine, CA cab. Too bad we don't have like Ubertine, right? But the old days. I took exactly one cab ride when in the nearly 20 years I spent in Morgantown, West Virginia. It was not a good cab. But anyway, the cab just kind of drives it in, drops it off, goes back out. All right. But the key is we have two different transferases because we have two different mitochondrial membranes. So once we get our fatty acid in there, we can oxidize it and generate energy. And this is going to occur in 3 stages. So stage 1, we're going to convert into 2 carbon units. So we're going to snip off 2 carbons at a time. All right. So sometimes when I'm bored and sitting out waiting for my kids to get out of school and pick them up, I'll watch like the little Facebook videos or something like that. It's like, oh yeah, look at this amazing technology, and they'll show like people making tennis shoes, and they have like the well, I'm not. This doesn't work, but like you think of like Keds or something like that or Converse that have the little rubber bit that wraps around it. So they really have this giant string of rubber in there, and they just cut off a section that's enough to wrap one shoe, and then they cut off another one for the next shoe, and cut off another one for the next shoe, and so on and so forth. That's essentially what we're doing with our fatty acids. Our whole fatty acyl CoA is our big long chain in there. We're just going to cut off what we need each time and make acetyl CoA. Okay. Stage 2, once we have our acetyl CoA, we're going to feed that into the citric acid cycle. So we talked about that in chapter 16. Yay, we got that. And stage 3 is now that we've made all this NADH and FADH2, that's in the electron transport chain. And yay, you already had your exam on that, right? Sad thing is, we're doing this backwards, right? We talked about stage 3 first, and then stage 2, and now we're talking about stage 1. So 3, 2, 1, blast off. So let's talk about making the actual acetyl CoA in here. Okay. I like this picture because I think this visualization actually helps students understand. Here we have, palmitate. Okay, so we got I think 16 carbons in here, and if we cleave these off 2 at a time, we're going to make 8 acetyl CoAs. Okay, so we can run the citric acid cycle 8 times, and from that we're going to get 16 carbon dioxides. Okay, because we got 16 carbons, 16 carbon dioxides, but we're also going to transport 64 electrons to make NADH and FADH2 and get those into the electron transport chain. And I'm pretty sure there's a question that comes up. We may do it as an example. It's like, well, how much energy do you get out of one, palmitic acid in there? I think sometimes on the take-home portion of the exam, I've picked a random acid and say, how much energy would you get from this molecule? How many ATPs would you make? And there's never a purely correct answer. There's a range where that goes in there. So if I do opt to include the question like that on the next exam, it doesn't have to be exactly right. And if you start looking it up, you will get several different answers in there. What I'm looking for is, well, how many acetyl CoAs are you going to make? If it's an odd chain fatty acid, because what happens, 16, we just cut it 7 times and we get 8 2-carbon units in there. What if we have like a 19 carbon, and you got 3 in there? What happens there? And it means we have other enzymes that require a little more initial energy in there. Okay. Anyway, we got 16 carbon fatty acid. We've attached it to coenzyme A. What happens? We have a dehydrogenase in there. Okay, so we're going to end up making this nice little structure here, where we have created a double bond over here, all right? And then we can oxidize this double bond, add a hydroxyl group here. So now we have this nice little section here. We can, oxidize this again to get a nice little double bond here. And that will allow us to cleave this carbon-carbon bond to give us the acetyl-CoA, and we can replace that carbon with another coenzyme A group in here. So this is essentially how we cleave off 2 carbons at a time. We oxidize the 3rd carbon away from where the coenzyme A is bound, and then we just repeat that process until we've made all the 2 carbon subunits that we can. In this case, 8 for palmitoyl CoA. Okay. So if we go through these steps, we're going to dehydrogenate the alkane to an alkene, take away hydrogen, make that double bond in there. Okay. This is catalyzed by acyl-CoA dehydrogenase on the intermitochondrial membrane. And there are various isoforms of this acyl-CoA dehydrogenase. This is just more, I'm not going to ask you specific about this. I don't, well, no. Huh. Maybe I'll have a take-home question about this. I can change this up a little bit, yeah. But there are various isoforms, one for long chains. So if we have 12 or more carbons, that one works on it. Once we get it down to medium chains, 4 to 14 carbons, a different acyl-CoA dehydrogenase takes over. And short chain, once we get down to 4 or 8 carbons, we get short chain. But the key here is we have this nice little trans double bond that does this. And this is very similar to succinate dehydrogenase reaction in citric acid cycle. It's the same mechanism. We're just working on a larger component there. This does require FAD as a cofactor. And you can see our flavin adenosine dinucleotide and flavin mononucleotide in here. This is going to help because we can transport electrons through here through our conjugated ring structure and stabilize radicals of our semiquinone form. All right. The second step, we're going to hydrate that alkene. So now that we have a double bond, that's a good site for chemistry. We're going to hydrate it and add a hydroxyl group to that carbon 3. That's awesome. Great. Hooray. Then we're going to dehydrogenate the alcohol. So we can use NAD as a cofactor. And as we generate NADH, that will feed into various components as well too. And then finally, we are going to, now that we have that carbonyl group at carbon 3, we can break the carbon-carbon bond and generate our acyl-CoA. And this is an acyl-CoA acetyltransferase, sometimes called a thiolase in there. But the net reaction is we cleave that carbon-carbon bond. So we've shortened the fatty acyl chain by 2, where we've made this new carbonyl group. We attach a new coenzyme A, so we just have a fatty acyl CoA that's 2 carbons shorter. And then we repeat that again over and over and over again until all the carbons are gone. All right. So the fatty acid oxidation is performed by a single trifunctional protein. So I think I mentioned a week ago or so, something like, hey, we're going to talk about the trifunctional protein. All right. So we have this lovely little octamer in here with some alpha subunits and some beta subunits. And the neat thing about this is it will allow substrate channeling between the enzymes. So as we do each reaction, our product of the first reaction feeds directly into the second reaction, into the third reaction, so on and so forth here. And this trifunctional enzyme tends to be associated with the intermitochondrial membrane, loosely bound maybe, but associated in there, somehow attached or co-localized. But it processes fatty acid chains with 12 or more carbons, and the shorter chains can be processed by soluble enzymes. All right. So when we start with our typical big fatty acid, it's right beside the membrane. As the fatty acyl groups get smaller and smaller, they can be solubilized. They will dissolve then, okay, in essence. All right. So our trifunctional enzyme. We're not going to talk about the gram-positive bacteria, but short chain specific system is in the mitochondria in there. I'm more interested in that you get the first part here, because all these are essentially the same type of deal that's going on. It's just our trifunctional enzyme is in the mitochondria to start with. And we have all other stuff there. Anyway, each round through, we shorten that carbon chain. So the first round, we're left with 14 carbon chain, then 12 carbon chain, then 10 carbon chain, then 8 carbon chain, 6 carbon chain, 4 carbon chain. And the last one, we end up with two carbon chains. So we end up making 8 acetyl CoAs. Just like if you were an awesome ninja and throw a stick up in the air and cut it 7 times, you get 8 pieces. All right. So for palmitic acid, our simple saturated 16 carbon fatty acid, that is what we as humans can make. We repeat this step 7 times, gives us 8 molecules of acetyl CoA. We get an FADH2 for each cycle and an NADH for each cycle. So we make 7 of those. And of course, the acetyl CoA can go into the citric acid cycle. So we can make more GTP, more NADH, and more FADH2 from the citric acid cycle. And all those electrons, which we kind of already totaled up, 64 electrons, will enter the electron transport chain. So if we look at this, how much energy do we get? All right, so beta oxidation, 7 FADH2s. Typically, you get 1.5 ATPs for each the FADH2, so that's where the 10.5 comes from. You get about 2.5 from each NADH, so that's where the 17.5 comes from. The citric acid cycle, we have 8 acetyl CoAs, so we're going to get 24 NADHs and 8 FADH2s in there. Okay. So that means typically we're going to see about 20 for each of those NADHs. 2.5 times 1.5 times will give us 12. If we look at the succinyl CoA synthetase in here, we directly make GTP. GTP is essentially the same thing as ATP, so that's why we include that over here. We add all this together, and 1 molecule of palmitic acid can potentially make 108 molecules of ATP. Okay. So there we go. That's our rationale for this. So this is, I'm not going to require you to memorize how many ATPs do you get for each FADH2 or NADH, but this is a good chart to kind of keep in mind to show you where this stuff is coming from, just in case I do say, you have, arachidonic acid that you, completely oxidized. How much ATP are you going to get from that, and why? So, and it's something like this that I want to see the why. That all makes sense so far? Nothing. Yeah, no,
Yeah.
I get one head bob, one head shake. I'm like, yeah. it's, it's always challenging to figure out how much I want to talk about each particular process. Because if we go back up and start looking at the trifunctional enzyme, we can talk more about the binding and more about regulation and more. And we'll talk a little bit about regulation and more about genetic transcription and so on and so forth. But I really just want to kind of focus on the overview, because we could have an entire course on pretty much each section that we talk about here. This is our introduction. Yeah, good times, right? And then I was about to say the good news is, but I don't know if it's good news or bad news, because if you're trying to learn stuff, if you look at other pathways, they have nearly identical mechanisms to achieve the same thing. So here we are making our acyl CoA. Here we are making our acyl CoA, and here we are. Yeah. So if you know one pathway, you know three pathways, whether it's beta oxidation of fatty acids, or running through the citric acid cycle, or oxidizing, say, isoleucine or leucine or valine as an amino acid. Same mechanism. You're generating FADH2, you're generating NADH. So it's just different enzymes are binding these. You can essentially say they're different flavors of enzymes that are performing the same mechanistic reactions to give you the same, well, not exactly the same products, but mostly the same products. Here we have another fatty acyl CoA. Here we have an acetyl CoA. Here we have something else that's similar to that. So if you know one, you know all three, but all three are different pathways, and then keeping the different pathways straight sometimes becomes a challenge, because this all can blend together. It's like, well, we're doing, this enzyme catalyzes this reaction, but it's in three different pathways. Or this very nearly identically named enzyme catalyzes the same reaction. So, and that kind of comes down to, if you know one of these pathways, you know all of them. It's just keeping them straight, right? And if you do that through purely mental recall, that's actually awesome and amazing. But what I'm going to stress for here is just kind of the application and being able to go to the source material and figure out, oh, okay, if I have a mutation here, maybe I'm going to see, yeah, that actually might be a good question. If we have one of these mutations in here that causes a particular disease, and say, well, you see a similar mutation in this pathway, what would you expect to happen? And you can say, well, it's going to be similar to this one, where we're going to make too much of this product and we see a buildup of something, or something. Rationalization. All right, so that's for palmitic acid. All right, that's just our basic fully saturated 16 carbon even numbered fatty acid. What happens, because we know a lot of the fatty acids that are stored in triacylglycerols are saturated and have double bonds in there. Okay, so these cis double bonds, these are not a substrate for the enyl CoA hydratase in there. So we need to add a couple more enzymes to account for those cis double bonds. All right. So we have an isomerase, which will convert the cis double bonds to trans double bonds, and a reductase that will reduce those cis double bonds, not at carbon 3, to saturate the rest of it. Okay. So if we have a mono unsaturated fatty acids, we only need the isomerase. All right. So we got that carbon 3 there. We can convert that and we can deal with it. Polyunsaturated enzymes are going to require both. Polyunsaturated fatty acids are going to require both enzymes, one to take care of the one at carbon 3, and then the other one to take care of all the rest of them in there. Why couldn't you just cleave those off and then move down until it's carbon 3? Apparently, that was not the way we evolved. So I was going to say Jesus didn't like it that way. So here we see what happens, with, say, oleoyl CoA, an 18 carbon monounsaturated one. We go through the first 3 steps, just normal as can be. And then we get down to here, and now we have this cis double bond at carbon 3. Our isomerase is going to convert that to a trans double bond, and then we'll be able to go ahead and cleave it. Okay. Why is a trans double bond okay? Why don't we need another isomerized another enzyme to get rid of it? We already have one. All right. Let's go back up here. Here we've basically made a trans double bond from our acyl-CoA dehydrogenase. So the mechanism, the enzymes for that mechanism are already in place. Okay? So they're working on the trans double bonds that we've made from the dehydrogenation in that first step. Okay. That's a good question too. Yeah. All right. But the key is we need an isomerase to convert the cis to trans. Okay. If we look at something like what do we have here? The oxidation here. Linoleic acid. Obviously, my name is covered up here. So linoleol-CoA gets formed in here. We go through our first three reactions, normal, chop, chop, chop. Then we get to this, carbon 3 has a double bond. So we have our isomerase that comes in here and converts that into a trans double bond. But we also have a reductase that is going to convert that into a saturated fatty acid. But what happens is for each one of those that we have, it requires some energy in there. So we're going to produce one fewer FADH2. Okay. But we make one during the first step of the next cycle. So it's not like we're losing much in there. And NADPH is going to reduce these remaining ones. So there's no further loss. Oops. All right. So first double bond requires isomerization. Second double bond, we have the reductase. Okay. And then isomerization. Alrighty. All right. So that's if we have even numbered unsaturated fatty acids. Let's make it worse, right? What happens if we have odd numbered fatty acids? Because we're just, going 2 by 2, all right? At some point, if we have an odd number, we're going to have 3 carbons left. So we're going to make a propionyl-CoA, which will be our last component. So when we cleave the last one, we're down to, if we start with like 19, chop, 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 we get down to 5, we make an acetyl-CoA, and we're left with a propionyl-CoA. Okay. There are also some bacteria that will produce propionyl-CoA as well too. Many plants will make this as well too, but propionyl-CoA will then be oxidized, and we can add a carbon unit to it. So we're going to basically, add a carbon dioxide coming from bisphosphate in here. Again, you see biotin as a cofactor that's going to be hanging on to that, essentially that carbon dioxide and bisphosphate in there. Yeah, yep. And that may maybe yeah. I'm not going to speculate. I was going to say, but bacterial metabolism in ruminants, it's like, I used to joke back before my facial hair started going gray. Dark hair, but I always have a few red hairs in there. Those are the ones that have turned gray. And we raised Angus cattle. They have black fur, but they'd always have a few red hairs in there. So I'm like, oh, I'm related to the cows, so maybe I'll get the. Unfortunately, I don't have a leaky gut syndrome or anything like that too. But again, you do see biotin prescribed for a lot of things for health, because it can help. In theory, if you have enough cofactor, the idea is if you have enough of it, it's going to help promote these single carbon transfers, which will help with skin elasticity and also. Yeah. So we add a carbon to it, and now we have a 4-carbon unit in this methylmalonyl-CoA. All right, and now we can, you can see we have a carbonyl carbon here. We can, kind of mutate this around, so we switch our coenzyme A from our last carbon to our carbon that we just added on here. I'm gonna say, well, really, that's that's not such a huge change there. But anyway, we go from methylmalonyl-CoA to succinyl-CoA is essentially what we end up making. And again, our vitamin B12, again, coenzyme B12 is important for this as well too. So we basically take our propionyl-CoA through a couple steps to convert it to succinyl-CoA. Hey, that's in the citric acid cycle, right? So there you go. All right. But you'll note that this does require ATP. So we have to spend some ATP to power this up to convert our propionyl-CoA to methylmalonyl-CoA. So when we add that carbon to our 3-carbon unit here, we are spending ATP to do so. Okay. And then once we spend the ATP, then we can kind of move this around. And it's origami. We make succinyl-CoA. Take a look at some. Need to check, make sure I'm not okay. Good, fine. Quick peek here. Oh Lord, okay. Oh yeah, okay. We're gonna do something fun on Tuesday then. I hope I'm gonna come up with something fun for you guys on Tuesday. Keep saying that. I will. Yeah. Yes, yeah, yes. That it was January 1st of last year, I think, that they stopped supporting Flash, and that's when all my links died. I need to check on that. I know the videos still work, but I'm not sure if the Flash. Yeah, I'm not sure if I have any Flash for this one, but I'll do that right after this class. I'll sit over there and figure that out. Yeah. I still have not heard from the bookstore, so yeah. Yeah. I will call them, but I did find out. I know for Trent, for the senior seminar, I just have not called them. But I know you're interested in Callidium. I need to call them and verify that we can do what we want to do. But I will send you a link for the volunteer form application, assuming that once I actually remember to call Callidium before 5 o'clock. It's like, oh, 5:30. Oh crap. Oh yeah. It's for the past 3 days. But I have an open link sitting in my tabs in there, so I need to do that. Anyway, one of the things that we saw, one of the cofactors is coenzyme B12, and essentially what we're doing is we're just kind of shifting our position over here one time. This mutase, we're moving our coenzyme A from one position to the other, and coenzyme B12 is really a good cofactor for doing that within these. Okay. Yeah. All right. Oops. Clicked on some things here. The reason that B12 is good at that is if you look at the structure here we have this lovely little cobalt in the center of this massive ring structure. And we've got, of course, a deoxyadenosine attached because everything has to have a deoxyadenosine in there. But we also have a nucleotide ring down here. So we have a lot of ways that we can stabilize and interact within this core and ring structure. Okay. We won't go into the mechanism of this, but vitamin B12, you will see that in a lot of these as a cofactor in a lot of these similar reactions. Okay. Basically, functional group exchange. All right. The reason I keep this slide in here is I like the drawing. All right. So as opposed to our structure that we have here, we can reduce this down to, look, here's a big square with a cobalt in the center, and we can change the oxidation state in here, right? Yeah, from cobalt 3 to cobalt 2, and that's what allows this to be, a cofactor, because that changing oxidation state, just like we see with iron 2 and iron 3 for oxygen transfer in red blood cells. Is that what it was? Yeah. We can do something similar with here, and the fact that we have this big core and ring with all this stuff attached to it allows us to kind of move functional groups in there. I will not go into this much detail to ask you about this stuff. Although I think this might be the exam where the take-home portion, it's, I have a question in there that's like, where in the world does this come from? How do we make the core and ring or something? What's required to make vitamin B12? But that's more of a hide and seek type thing. All right. I just want to mention beta oxidation primarily occurs in peroxisomes for plants, but we're not going to deal with plants, people only. But there is a nice comparison here. So if you happen to have one of the biology classes at the same time, you can look at the mitochondria pathway versus the peroxisome or glyoxysome pathway and look at the similarities and the differences in there. Okay. But there is a lot of overlap in the beta oxidation of fatty acids. The last part of this chapter, we can talk about ketone bodies. So we saw from chapter 16 that when acetyl-CoA goes into the citric acid cycle, it requires oxaloacetate. If oxaloacetate levels are very low, then we end up making ketone bodies instead of regenerating that oxaloacetate. So oxaloacetate gets depleted. Instead, we will make acetone, acetoacetate, and this beta hydroxybutyrate. Okay? These three particular ketone bodies can leave the liver, and they can be used as fuel by the brain in particular. Okay? And that's kind of the liver's job, is to make sure the brain is functioning. So that's why we see these ketone bodies made in here. We also get a lot of free coenzyme A, not free as in, it doesn't cost anything, but with nothing attached, coenzyme A in there. So you can see 2 acetyl-CoAs. We can have a thiolase that will release coenzyme A. And here we have our acetoacetyl coenzyme A. And if we throw in another acetyl-CoA in there, we can get the beta hydroxy beta methyl HMG-CoA. Right? We will see HMG-CoA later on when we talk about cholesterol metabolism. Woo, yay! So everything's interconnected, and everything is part of multiple pathways. But this HMG-CoA synthase also becomes very important as well too. But anyway, acetone, acetoacetyl, acetoacetate, and beta hydroxybutyrate are our three ketone bodies that we end up making. Okay. So in order to make these, our coenzyme A, if we want to get these ketone bodies to someplace else, we got to get rid of the coenzyme A. We will then cleave off the coenzyme A to have, through either the actions of HMG-CoA lyase, or then we convert acetoacetate to either acetone or beta hydroxybutyrate as well too. So you can see a lot of this is reversible. But these three compounds can be released into the bloodstream and can travel to other tissues. The interesting thing about acetone, it gets removed as a gas, and you can actually exhale it out. So what you may often see in some diabetic patients, if they start to go into diabetic shock, their breath smells like acetone. They are not, using glycolysis. So if one of the ways you can see if they're starting to go into diabetic shock was if their breath smells like fingernail polish remover. That's the acetone that's being exhaled in there. Okay. So again, the liver is the source of the ketone bodies, and then they travel throughout the bloodstream to get wherever they need to go. Okay. So in this case, we normally send glucose to the brain and other tissues, but if we lack oxaloacetate, then instead we get the ketone bodies, and they can be exported to the brain and heart, muscle, kidney as well too. But we want to make sure the brain is still functioning in there. When these things are made as fuel, all right, we should we go in here? Ketone bodies as fuel. Okay, I already kind of said that. All right, build up ketone bodies though. We will talk about diabetes, I believe, in the next chapter as a model disease. So understanding diabetes helps us kind of understand how this, how the flux through these different pathways works under normal conditions, and then doesn't work during disease states. We also get into, one of my favorite questions is like, what happens if you change your diet here? So the Atkins diet or the no carb diet or something like that, it actually worked out really well. Biochemically, it's great. We kind of, inhibit, stop eating a bunch of sugar. Got to get rid of my Coca-Cola, and we will burn fat and protein for energy in there. That works great. Worked great for a lot of people because most people cheat and will have a little bit of carbohydrates. So you still have some that's going here. If you completely cut out sugar, especially if you go cold turkey, then your body's not getting enough time to adjust and you start making ketone bodies. And there is a chance that you can experience ketoacidosis. And there were people who would just absolutely eat no carb, must have no sugar, obsessively about that. And some of them would run into health problems. And I have a question that kind of asks about a dietary change for some people with some conditions or something like that. And I want to see, there's not a correct answer. I just want to see the thought processes that go through that. It's like, is this a good thing, a bad thing? You can, and there's often an adjustment period. If we go back to World War II and the liberation of concentration camps, the Allies rolled into a couple first concentration camp, and there are all these emaciated Jews who were just completely underfed, and they basically had like thin gruel with just a little bit of carbohydrates for months or years. They're like, oh my God, let's give them steaks and candy, and a lot of them died because their bodies had adapted to not have that. So suddenly they had a lot of other food in there, and there was a buildup of metabolites, some of which couldn't be cleared fast enough and became toxic. So I'll have to look up exactly what killed them, but it's like, whoa, okay, we can't just feed you a steak if you've been starving for a year or something like that. So yeah. So anyway, this is chapter 17. Fats are a huge energy source. 2 carbon units. We basically go 2 by 2 by 2 by 2 for everything. We kind of looked at palmitic acid as our initial model. Then we looked at what happens if we have an unsaturated acid in there. We need 1 or 2 more enzymes, depending on if we have 1 double bond or multiple double bonds. What happens if we have an odd number fatty chain acid? We make the propionyl-CoA, which ends up giving us HMG-CoA. And then, of course, our ketone bodies, knowing acetone, acetoacetate, and HMG will be, those are the ketone bodies that can be used. And we'll see more about that coming up here in 18. Yes, in fact, I'm pretty sure that exam 2 will have a substantial take-home portion. So like the in-class portion might take you guys half an hour, and so I just can't make a long. Yep. Yeah. 16, 17, 18. Oh, so next up is urea, and then we did 19, we did 20. We're skipping 20 because that's plants, right? I need to change the title of these so they actually tell me what they are. So, all right. Yeah. So I think this next upcoming exam is only 3 chapters, but the next chapter is actually 18. Yeah, 16, 17, 18. And then we already did 19. We're skipping 20 because 20 is all about plant enzymes. So then exam 3 is 21, 22, and 23. I believe that is going to be 21, 22, and 23. So yeah, that's where we start tying everything together. So I think exam 2 generally is not too horrible. Some of the questions are a little out there, but that's why it's a take home thing there too. And it's really the process. What's that? Oh no. Exam 2 and Biochem 1 is awful. Exam 3 was good. Yeah. I was gonna say I should stop saying that because I think people get too confident after exam 2 and exam 3. So yeah. Um, so the last thing I want to look at is that let me take a look. Oops, I should probably share this so you guys can see this too. Okay, so let's take a look at my helpful items for exam 2. Right, close that one down. Yeah. So slow. Ah, damn it. Okay, a couple of these are flash, but I know there are some short videos in here as well that will help for the exam 2 stuff. But I'll see if I can find some alternates for those. Crap. Gotta find a replacement for Flash or an update for these. It's a shame because a lot of this stuff was really good the way it would just allow you to kind of click through. And of course, this is going to be super slow for some reason. Anyway, I think that's all I've got for today. So